So the beautiful thing about the integument is that it is a highly mitotic layer and can give rise to lots of different structures by reorganizing the cells and forming particular architectures out of them, and then adding more and more keratin and intermediate filaments to those cells and cementing them very close together can make structures very hard. And in combination with connective tissues like cartilage or heavy collagen or even bone, then we can make a lot of different protective structures that are found on the surface of organisms. So in this particular bighorn sheep here, we have basically long horns that are permanent structures that are part bone and part skin. And then obviously fur here, right, or the hair that is a mammalian characteristic. This is a cross-section through skin, through what we're calling thick skin here. So you got a couple layers that have different colors. We're going to go through and talk about those layers as we discuss the integument. So the typical anatomy of the vertebrate skin includes two major layers. One's an epithelial layer called the epidermis, and the other layer is a connective tissue layer called the dermis. Epi means outside or on the outside of the dermis, and dermis is the deep layer. The dermis is typically relatively thick in many vertebrates and will be kind of the glue that holds the epidermis to the underlying tissues. And also it embeds and cements into place a lot of these really large structures that are produced by the skin. This is the figure you see in your text, and this is a drawing that I pulled off of the internet. That shows you epidermis and then the dermis. Often there's a hypodermis underneath. The hypodermis hypo means below. Hypodermis is not typically thought of as a skin layer, but is often the layer that's called superficial fascia in some gross anatomy textbooks, and is often a connective tissue that's collagen-based or is adipose tissue in mammals, for example. The dermis can also have a lower layer or the deepest layer that's called the stratum compactum, or the dermis can be called the stratum compactum in certain textbooks. So particularly during development, the dermis develops as a layer called the stratum compactum. The epidermis will develop on top of that, and the epidermis during development is called periderm outside of the dermis. So the epidermis is derived from ectoderm, our outer layer of embryonic tissue, and is a stratified squamous epithelium for the most part. It can be thin, a couple layers thick, or very thick, 30 or more layers thick, depending on what organism we're talking about. The dermis is derived from connective tissue. It's actually mostly derived from somites, which are balls of connective tissue, developmental cells that form around the developing spinal cord during development. I'll show you pictures of that later. There's a basement membrane between the epidermis and the dermis that is produced mostly by the basale layer, or the base layer of epidermal cells. Again, I talked about the hypodermis is deep to the dermis and is typically made up of connective tissue, sometimes bone, and often, often adipose tissue in mammals. It's also called superficial fascia, as I've mentioned before. Again, on this particular image, we have some beautiful structures that are produced by the skin. The shell here of the turtle is a composite structure. It's part bone and part skin derivatives. The bone itself comes from two sources. It comes from the dermis, and that part of it of the bone are called scutes, which are on this slide here, the dermal bone that's formed. And also part of the bone is the ribs and the vertebrae, which are part of the endoskeleton. But the epidermis is really the productive layer. It produces a whole bunch of different types of structures for organisms that help with their interaction and protection from the environment. Things like quills, nails, talons, hair, baleen, claws, horns, beaks, and some scales are all epidermal derivatives. The dermis also helps produce some of these structures. It produces dermal bone and also some scales, particularly fish scales. So notice scales can be produced by both structures, the epidermis and dermis. Typically the more ancestral scale, the fish scale, is a dermal derivative. It's actually the leftover or remnant of what dermal bone that first developed in the ostracoderm fish. Whereas the scales in tetrapods, particularly amniotes, birds and fish, and some mammals, are epidermal derivatives. So Again, the epidermis is really, really good at this because of mitosis. Mitosis produces hundreds of thousands or millions of cells that you can then organize into specific structures. Also, keratin is the major product that is produced inside of these cells. And the keratin, remember, is a type of intermediate filament, which makes the skin cells very hard and tough and flat. Here are some functions of skin. It's a critical barrier between the external and internal environments. It's actually a perfect barrier, if you will. And its mitotic ability means it's shed often in many vertebrates. It is part of the exoskeleton, that is the outer barrier of the body. Even though you don't think of vertebrates as having an exoskeleton, it kind of replaces what you would consider exoskeleton in insects and other invertebrates. It's used in gas exchange often, and osmoregulation. A lot of skin like ours is waterproof, all right, and prevents water from leaving the body. Used in gas exchange, osmotic regulation, the skin gathers heat and radiates heat, so it can absorb heat and get rid of excess heat. We have specialized glands in our skin called sweat glands, which actually evaporate water off of the surface of our skin and draw heat from our bodies. It holds all of these cool structures that not only forms them, but holds and cements them in place, things like feathers for locomotion, nails, horns, and whatnot for protection. The skin also produces a number of glands, which I haven't even started talking about yet, which are involved with maintenance of the skin and protection, and also for moisture. Color is also incorporated into the skin in a lot of organisms or into the structures that the skin makes, which allows for recognition and reproductive purposes. And it prevents water loss and penetration of invaders such as bacteria. So these are all kind of major functions of the skin, if you will. Okay, we talked a little bit about the embryonic origin of the skin, but let's show you a couple different pictures here. So over here is a gastrula, a developing gastrula. Just to remind you, if you haven't had a lot of embryology. So when sperm and egg come together, that forms a zygote, right? A diploid zygote in vertebrates. That zygote's going to undergo rapid division, right? During development, and that rapid division is called cleavage. All right. And so cleavage is going to produce a ball of cells that is called a blastula. All right. This blastula will be a hollow ball of cells filled with liquid. The blastula then will buckle on one side. So you get this hollow ball of cells. That hollow ball of cells will then buckle on one side like this and form the structure we call the gastrula. And so up here is the gastrula, a picture of a gastrula. And what happens are some of these cells are going to be pulled inward as they buckle, and they're going to form a new tube. That new tube that develops here, that's developing, is called the archenteron. All right. So this is called the archenteron. All right, the archenteron, and the hole here is called the blastula, or blastopore. Sorry. It's called the blastopore. All right. And this archenteron becomes the digestive tube. It will continue to grow until it goes to the other side of the gastrula and pierces that size. And then you have two openings, right? One will be the anus, one will be the mouth. And then the lining of this tube, the lining of the inside tube is, as you can see, here's kind of a drawing up here in yellow, will be endoderm. And then the layer of cells on the outside is called ectoderm. And then some of these cells will differentiate from the endoderm and move to the middle and fill in the space between the endo and ectoderm, called mesoderm. So the skin comes from mesoderm and ectoderm. So the deepest layer of the epidermis forms what's called the stratum basale layer. And that's going to be formed like two layers of cells. So there'll be a stratum basale, and then it will divide by mitosis and form a very flat group of cells on the outside of the developing embryo. That's called periderm, which is going to eventually divide and continue to grow and become what we call epidermis. And underneath the epidermis, we have the development of connective tissue that's really compacted, all right? That's really condensed, and that's called the stratum compactum. All right, so the stratum compactum will become the dermis. There are several cells that arise from what are called neural crest cells that come from basically an area between what are called somites and the developing nerve cord. The cells that give rise to the stratum compactum come from the somites. So let's talk about somites. So somites, what somites are basically groups or balls of cells that come together and they develop as kind of staging sites, if you will, for connective tissue cells. And they develop or are organized by mitosis and from migration right next to the developing spinal cord here, which is labeled N. And then they will then migrate out to all kinds of areas within the body. One of the areas they migrate to is the stratum compactum, and they form the stratum compactum and become the dermis. The neural crest cells are the cells that are found between, right in this area here where my pointer is, between the somites and the developing nerve cord. And they give rise to specialized cells. Some cells that come from the neural crest area are chromatophores or melanocytes, which are the cells that give pigmentation to the skin. So that's a little bit about the development of the skin and some of where these structures originate from. So let's talk about the dermis and epidermis individually, and then we'll do a comparative review across taxa. So the dermis in many vertebrates is I should say in all vertebrates, is the connective tissue layer, right? And so it sits underneath the epidermis. This is a picture of thick skin, like at the heel of the foot, for example, showing you a really thick, dark red or pink area, which is called the epidermis. And then this lighter area underneath is called the dermis. All right, and so the dermis can often give rise to bone, all right, what we call osteoderm. So osteobone, derm is skin, meaning dermis. And this type of bone formation is called intramembranous bone formation. So intramembranous, which means it is going to develop within a membrane. The membrane here, intramembranous ossification, is the dermis. The dermis is made up mostly of collagen fibers. That's the major protein produced by the cells of the dermis, which are called fibroblasts. All right, and the collagen is in the formation of what we call dense irregular connective tissue. That is, it's just laid down in a bunch of wavy, random kind of pattern that give strength to the dermis, and also the dermis is very sticky because the fibroblasts produce a lot of what we call glycoproteins, sugar proteins that attract water and are very sticky. In fish, the dermis is much more organized. The collagen will be organized into bundles that form really large layers of very uniform collagen fibers that are called plies. And then those plies are going to be organized into kind of a web that stitches the dermis to the epidermis, and we call those a bias. And in fish, that's what makes it really hard to skin the shark, right? Sharks are fish, right? Is the collagen underneath. And what these bias do is they prevent the skin from wrinkling when the fish is alive, so that it reduces friction when they're swimming in the water. Our skin is much easier to remove because we don't have to worry about friction in the air. The air is less dense, it's easier to move through. So that's a little bit about the dermis. We'll talk more about the dermis as we move into talking about structures. The epidermis is a stratified squamous epithelium. The layers protect the underlying tissue, forms a beautiful boundary. The layer that's closest to the dermis, or the deepest layer, is called the basale layer, and it is the mitotic layer, produces tons of cells. It's always undergoing mitosis. And this will then, new cells will be pushed up, and the oldest cells will be shed. So in most vertebrates, the skin is continually shed. In our skin, our thickest skin can be many, many layers, 30 to 50 layers thick of these cells. The cells are called keratinocytes. Keratinocytes because the cells are in the process of making keratin, at least in terrestrial vertebrates. And keratin cornifies or hardens the skin and waterproofs it. Amphibians are the only vertebrate with capillaries in their skin. They actually have blood vessels in their skin because they respire through their skin. We don't have capillaries in our epidermis because we don't respire through our skin as a major respiratory organ. Most of the capillaries are going to be found just underneath the skin and the dermis and in a region that are called dermal papillae, which are kind of the basically upward movement of the dermis into the epidermis, particularly in thick skin, which will deliver blood vessels up closer to the deeper layers of the epidermis and provide them nutrients. Because blood vessels are restricted to the dermis, these cells are the furthest away up here are going to die or be dead cells because they're too far away from their lifeline or their blood supply. This process of accumulating keratin and dying and then being shed is called keratinization, which you can see here on the bottom. The outer layer of the cells is called the stratum corneum because the cells are flat and hard and they flake or are going to come off. And so it's all right, let's talk about chromatophores or melanocytes. All three of those terms, chromatophore, melanophore, or melanocytes, for us will be synonymous. These cells in all vertebrates produce color, right? And they do that through the production of melanin. There are a whole bunch of other colors that can be made outside of the melanocytes. We are going to focus just on the melanocyte, which can darken the skin and add color to the skin. There are different types of melanin. There's eumelanin, which is kind of the dark brown color that gives us a tan. There's phyllomelanin, which adds more kind of red or orangish color to the skin. There's also keratins or carotenoids that can be concentrated in the skin. But for melanin production, let's focus on the melanocyte here. And it's pretty interesting how melanocytes make melanin. I'll go over it really quickly. Typically, chromatophores are these melanocytes are going to be found either in the basale layer, all right, the most basal layer of the skin, or just underneath the epidermis. And it depends on the vertebrate. Most, for example, amphibians and fish have the melanocytes just underneath the epidermis, whereas a lot of tetrapods, particularly mammals, have the melanocytes inside of the basale layer. And what these melanocytes do is produce melanin, and they have lots of cellular processes that come off of them and kind of spread out within or around the basale layer cells. And what these cellular processes do are deliver melanin granules to the cells. The melanin is actually exocytosed to the interstitial fluid or interstitial space around the basale cells, and then the basale cells take them up through phagocytosis, and the melanin granules are then delivered to right above or right on top of the nucleus, forming what is called a melanin cap on top of the nucleus. And these melanin caps, as you can see in these cells here, basically absorb the UV light coming in from the sun and prevent damage to the nucleus, which is really important because the basale layer is the layer undergoing mitosis, right? So because it's undergoing mitosis, it is very sensitive to DNA damage as you condense the chromosomes and whatnot. So you protect those cells with melanin sunglasses, if you will. Now, the process of melanin production is pretty interesting. Melanin is a derivative of a single amino acid that's called tyrosine, all right? So tyrosine is the amino acid that we're going to use here. You can see down here. So tyrosine is going to be taken up by these cells. And what happens here are the genes responsible for melanin production, the main one being tyrinase, which is the enzyme that does this. It is going to be basically produced as a messenger RNA, right? So you're going to copy this gene through transcription in the nucleus. The messenger RNA leaves the nucleus of the melanocyte, goes out to the rough endoplasmic reticulum, where it is translated into a protein that is going to be transported to the Golgi apparatus via a vesicle, transport vesicle. Once in the Golgi apparatus, the tyrosinase is going to be basically packaged with a bunch of other proteins that will then be released as a really large vesicle called a melanosome. And the melanosome has stage numbers 1 through 4. 1 being the immature, 4 being a mature melanin granule. And what happens is this enzyme is going to convert tyrosine into melanin. And basically, it's a process of hydroxylation, adding OH groups to tyrosine to form melanin. And that happens over time and accumulates on the protein fiber matrix that's laid down in the melanosome. And so that process takes time. And as the melanosome develops into a melanin granule, it will hop on microtubules and will walk up or follow the microtubules up into the cellular processes during the maturation process. And by the time the melanin granules mature, it'll be at the top of the processes and then dumped into the interstitial space. So that's kind of how it works. These are our 4 different looking melanosomes. Here's a stage 1 melanosome where you don't have much darkening and not much melanin being produced. Stage 2, you can see the protein fibers or matrix that are laid down. And then the melanin accumulates on top of those protein fibers that are inside of the melanosome as tyrannosase is increased in its activity. It will lay melanin down and become very, very dark as melanin is developed or metabolized from tyrosine. And your mature granule is a stage 4 granule. And that is what is released to the environment and taken up by the basali cells to form that melanin cap. So that's a little bit about melanocytes and the process of melanin production. Again, just some more pictures here. So these are the basali cells. You can see all of these dark granules. All these dark granules were taken up by endocytosis. These are melanin granules that are covering the tops of the nuclei. You can see really nicely, protecting them from UV damage. And again, you can see all this melanin accumulation where these red arrows are that are sitting on top of the basali cells. So the cells are completely covered, right, to protect them from UV damage or radiation from the sun. All right, so let's go through a taxonomic introduction to the skin. We'll start with our what we call ancestral or primitive, right? Vertebrates first. So let's look at lampreys first. And again, you've seen the lamprey skin before in lab. Here's a nice low power picture of the lamprey skin. Here is a picture from your textbook showing you the epidermis, the dermis, and the hypodermis. So hypodermis has a lot of fat in it and some really large veins. The dermis is highly organized and deplies so that the skin does not wrinkle during swimming. This is a scaleless fish, right? There are no scales produced by the dermis. The epidermis is several layers thick. You have epidermal cells kind of on the top. There's not a corneum here because the cells are not cornified. There's no keratin that's being processed in the fish skin. This is basically just a live epithelial layer that is several layers thick, and it has a very thick mucus on the surface of it. There are 2 types of cells here other than epithelial cells. There are club cells and granular cells. This club cells release chemicals to the environment when alarmed or excited, and the granulated cells are producing the mucus. So you can see the granulated cells here versus the club cells. So the granulated cells produce tons of mucus, as do the epidermal cells. Many of the cells here produce mucus, and remember, lampreys and hagfish can produce tons of that mucus, right? And form a very thick mucus layer that's very slippery and nasty, so that it's kind of their protective function. So that's the lamprey skin. So it's a little different than what we would consider, vertebrate skin or fish skin that has scales. Let's do shark skin next. So shark skin has scales. These scales are kind of hard bone or remnants of bone. They're called placoid scales, as you remember. And this is a picture looking down on shark skin. It's seeing the individual points or apices. Or the keel is another term that they use for the placoid scale. These are cross sections of the skin. This is a low power cross section of the skin, sagittal section of the skin, I should say. This is a high power sagittal section of a scale. We have the epidermis on top, which isn't very thick, right? And then the dermis, which is really thick, and then skeletal muscle underneath. The dermis again is organized into plies and bias, all right, to prevent wrinkling. The epidermis is several layers thick, and the placoid scale is produced by the dermis and actually penetrates or breaks through the epidermis. So the point or keel of the scale sticks out and gives the skin of sharks a rough texture, as you know, right? A sandpapery kind of texture. If you look at the scale, it is a tooth, or what we consider a tooth. The teeth in sharks look a lot like their scales, are just larger and harder, have more calcium deposit associated with them. But the placoid scale is hollow. It has a cavity in the middle of it that's called the pulp cavity. And inside of that are odontoblasts, which are cells that are tooth cells, bony tooth cells that produce the connective tissues of the scale. The scale has a very thin layer of enamel on the outside of it, and then a thicker layer of dentin, which is a spongy bone, and is they're slightly flexible, but they are hard and strong. If we look at bony fish skin, it's a little bit different because the placoid scale is not there. They have a different type of scale. There are a couple different types of scales in the bony fish. I'll talk about them in a minute. But the skin itself, again, is unkeratinized. It's several layers thick, the epidermis is. And then the dermis is going to be a thick layer that also holds the scales, as you can see here. Here are two really long scales. The scales are produced by the skin. They're basically compact collagen together, and then some bony tissue in many cases. So this is kind of a specialized osteoderm, if you will, or I like to think of as a reduced osteoderm, as far as what the bone looks like. Scales can be really flexible or really hard depending on the amount of bone that's there. So this is a higher power image of the skin showing you the epidermis and then the dermis underneath. This is a scale down here that has the spikes on it. It's a tenoid scale. This is the basal plate of the spike, of the spike scale. Up here is the epidermis, and these are goblet cells. Single-celled mucus-producing cells are called goblet cells, or they can often be called a club cell. All right, so club cell here is the same as saying a goblet cell that produces mucus, and it produces the mucus that sits on top of the epidermis, which protects it and makes the skin slippery, so they cut through the water very well. They have a bias, again, to prevent the skin in the dermis from wrinkling. And so it stitches the skin very tightly to the underlying connective tissue. So this surface slime is the mucus that's produced by the goblet cells. And again, fish, osteichthyan fish or bony fish produce a lot of it. Now, there are different types of scales that are produced by the dermis, depending on the taxonomic category of fish. There are cosmoid scales, ganoid scales, and then two teleos scales that are called cycloid and ctenoid scales. Cosmoid scales are very rare. They're only found in Sarcopterygians, and they are the thickest of the scales. They're almost like bone, real bone. They basically have an enamel surface, which is the hardest substance found in vertebrates. And then they have two layers of bone. They have dentin and vascular and lamellar bone. So this dentin layer, again, is kind of a more reduced bone. And then they have true vascular or lamellar bone, what we call spongy bone, that make up the scale. So the scale is quite thick, not flexible, and very hard. The ganoid scale is found in gar and some of the other paleonysiform fish. It has enamel, again, on the surface, and no dentin, so we remove the dentin layer. And then we keep the vascular and lamellar bone. So we're just removing the dentin. The specialized bones here or the specialized enamel here is called ganoin, which is how the enamel is organized. In the cosmoid scale, the dentin is called cosmin, right? Which is where the cosmoid scale comes from. Ganoid scales lack that dentin and still have the enamel, and then the vascular bone underneath. Again, it's a thicker, non-flexible or not very flexible scale. Gar have very hard scales on their outside, very primitive. The teleost, what you're left with is just lamellar bone. It's not calcified. You remove the dentin and the enamel, and now we have a very flexible scale that allows the fish to bend their bodies much better and to move much easier or more freely, if you will, within the watery environment. There are two types of scales. There are cycloid scales where the lamellar bone is in rings or circles. And then you got the ctenoid scale, which has kind of a spiked border to it. So it depends on what fish group you're in. The scales are made the same way. They just kind of, the collagen's laid down a little differently. So I have a little table that kind of shows you pictures of the scales. So here is the cosmoid scale, what it looks like. It's very shiny and very thick. Here's the ganoid scale, which again is very thick and bone-like. Movement's very limited in these. It tells you it's cosmoid scales are found in sarcopterygians, lungfish, and coelacanths. The ganoid scales are found in gars, beachers, and reedfish, which are all paleonysoforms. And then your derived fish have these two scales. Here's the cycloid scale that has the rings of collagen, lamellar bone. And here is your ctenoid scale that has spikes on its surface and then this brush border or comb border on it. And so those are the two forms that are found in your more derived fish, which are much more flexible. So those are your scale types. All of those are produced by the dermis, as is the placoid scale. So all fish scales are dermal derivatives, as I've mentioned before. There are some derived fish or bony fish that lack scales. Here's a blobfish, one of the strangest deep water fish in the world. It's got a very slimy, mucousy skin and has no scales, so it's scaleless. Have a very flabby skin. Weird fish. I just thought I'd show you a picture of that. So there's a lot of aberrations or exceptions to the rules out there as far as skin goes. All right, so let's move into the tetrapods, talk about amphibian skin. So as we move on to land, the tetrapod skin, as we move and go over the different groups, we're going to basically keratinize the skin more and more and more until it becomes completely waterproof. Amphibians have a skin where keratinization starts. They have a thin epidermis though, and the keratinization is not complete. That is, there's a lot less keratin in the amphibian skin than in reptiles, birds, and mammals. But there is keratin there. That keratin kind of hardens the skin and strengthens the epidermis because we don't have bone in the amphibian groups, right? There's no bone, no dermal bone, no scales except in caecilians in the amphibians. So their epidermis is a little tougher, stronger than fish skin. Amphibians still produce lots of mucus, but the mucus isn't from a unicellular epidermal cell. It's not from a club cell or a goblet cell. They come from actually multicellular glands that are epidermal derivatives. And these are called mucus glands. So when you look at amphibian skin in sagittal section, you have an epidermis and a very thick dermis. And you can see you got these large round structures, which are multicellular glands. Some of these multicellular glands are actually mucus glands. And so the cells produce a lot of mucus. Mucus is basically a glycoprotein. The protein produced by mucus glands and by goblet cells and club cells is a protein called mucin. It's the name of the protein. And it's a glycoprotein. It's a sugar protein. And it attracts water. It's a water sponge, absorbs water. And so that forms the kind of slimy mucus that you see, the sticky mucus. So that mucus is going to be formed inside the gland and then travel up the duct and be dumped onto the surface of the skin to make the skin wet, moist, and slimy, as you find in salamanders. Salamanders also have a multicellular glands that are called poison glands, which produce products that either taste bad or acidic or are really toxic. And so we'll talk a little bit about those in a minute. These are exocrine glands. They are multicellular glands that dump into a duct. The ducts end on the surface of the skin. The dermis is a lot less organized. It's dense, irregular connective tissue, no plies, no bias, right? So the skin is wrinkly because we're moving through air now, not water. Some nice colored images here of different amphibians and their skin. Amphibian skin is fantastic. I've gotten into doing research on it lately, particularly salamander skin, which is less research than frog skin. I mean, they have incredible multicellular glands. So here's the image you see in your text, and these are just some microscope images from slides that have been made either by me or my colleagues. So you can see epidermis on top, dermis, There is a boundary or a layer here of chromatophores that are found between the epidermis and most of the dermis. The chromatophores here, or the melanocytes, are concentrated just underneath the epidermis, and they send their cellular processes up to the basale layer, again producing melanin to protect the cells and give color to the skin. This is a beautiful mucus gland right here that's clear and shows you the duct that dumps the mucus onto the surface. You can see how much larger the poison glands are, right? Here's a higher power image showing you the mucus gland. You can see kind of the mucus is flocculent or bubbly, and it's produced and dumped onto the surface of the skin. Here's the epidermal cells. There's a basale layer and then a corneal layer. And then they usually call this an intermediate layer of epidermal cells. They are called keratinocytes now because we're producing keratin. You can see the collagen of the dense irregular connective tissue of the dermis that the glands are embedded in. Here's a poison gland. You can see the poison gland produces protein granules typically, which are the toxins. And this is a part of a melanocyte here, or chromatophore, which produces the melanin. The other major cell type that you find in the epidermis are called Leydig cells. Leydig after the scientists who discovered them. They are white blood cells. They're basically macrophages that protect the skin from invaders. This epidermis often, particularly in salamanders, has capillaries in it, which is the only vertebrate that has capillaries in its epidermis. And those capillaries are really close to the surface because the skin is either the major respiratory organ in salamanders because they're lungless, or it is an accessory structure that they can utilize to breathe. Because as I mentioned, these poison glands can produce anything from a product that's kind of acidic that will burn your eyes or your mouth if it gets close to a mucous membrane, to something that tastes really bad, to alkaloids that are really toxic that mimic neurotransmitters like acetylcholine, for example. And some of these frogs, like this one here, Phyllobates terribilis, one of my favorite frogs, is one of the original described dart frogs that are coated by the indigenous people of Colombia, and other frogs in this group are also utilized this way by tribes throughout South America. They rub their darts on the surface of the skin, and these skin toxins cause paralysis and suffocation in animals or in humans. So these guys are left alone. Phyllobates terribilis is a very bold frog because it runs around, nothing bothers it because of its toxicity. It has enough poison that it can produce in a single spout of secretion that can kill up to 10 people. So it's one of the most toxic organisms on the planet. The alkaloid here is actually ingested, or at least what we need to build the alkaloid toxins, these frogs, is ingested with the insects, particularly ants and beetle larvae that they eat in South America. And they produce toxins that are called tetrodotoxins. The major tetrodotoxin in Phyllobates is called epibetadine, which has pain-killing properties. Matter of fact, its pain-killing properties are very, very, very good, if you will. They can kill pain about 200 times better than morphine does. So it's got tremendous potential in the medical field. The biggest problem is epibetadine also causes cardiac arrest. So they're working on trying to modify what this epibetadine looks like in the pharmaceutical industry so that it retains its pain-killing properties, but obviously doesn't affect the heart. If they can perfect that, it'll be a huge breakthrough, because as all of you know, morphine is very addictive if you take it too much, whereas epibetadine doesn't have these properties on the central nervous system that morphine does. But in any case, those are poison glands and how they work, and that some frogs produce pretty toxic poisons. As I've mentioned, the skin is an accessory respiratory organ. A lot of salamanders use it as one of, either the primary respiratory organ or as a accessory respiratory organ. So this is a hellbender here. These are two pictures of hellbenders. It's a completely aquatic salamander. It's the largest salamander in North America. They can get about 3 feet long. They live in clean mountain rivers. You can find them in Indiana, but they're very endangered. They're found particularly down in the Floyd Knobs region of southern Indiana is where there is a pretty stable population. In I think it's the Blue River system here in Indiana. But in any case, you can see these guys have lots of folds in their skin, and they like clean, cold streams because they can extract oxygen through their skin. They have lungs, but the lungs don't work very well, or don't work at all, really, in these completely aquatic salamanders. And so they respire almost exclusively through their skin using these wrinkles, which increase surface area. So they have cutaneous vascularization with the capillaries in the skin. Also, there are certain frogs that develop these hair-like processes. They're called hairy frogs. It's not real hair, obviously, but it looks like hair. And what it is just elongated papillae from the skin that are highly vascularized with capillaries. So you grow the papillae up, and then you get a cellular process or several processes coming off of the skin that increase surface area tremendously and allow these guys to extract more oxygen from the water. Males develop these to help with the metabolic demand of breeding during the breeding season. All right, reptile skin. So reptile skin, we get this greater commitment to keratinization. The skin becomes harder and tougher and thicker, therefore becomes less of a respiratory organ and more of a protective organ. We can also add bone under the skin, and bone is often quite common in certain reptilian groups to protect the organism from damage. A lot of times the scutes are found on the back to protect the animal from predation. Reptiles have some of the most waterproof skin in all of vertebrates and can occupy some of the driest places in the world because of the waterproof characteristics of their keratinization process. They also can produce epidermal scales, or folds in these scales, that are basically really, really tightly cemented and packed, highly keratinized cells that form the scales that you see on the surface. So this is a beautiful picture of really large scales that are made up of smaller scales. As you can see, there are kind of rhomboid scales in there, and smaller ones in the middle of the larger rhomboid scales. These scales can be granular, they can be triangular shaped, they can be rhomboid shaped. They come in a wide diversity of architectures, if you will. This is what a typical skin sagittal section in a reptile looks like. You've seen this before in lab, but what you're seeing here is the corneum. The scale here, the epidermal scale, in most reptiles is a corneal, stratum corneal specialization. And you can see the stratum corneum is very, very tight and cemented together to form that cap, which the point here is called the keel. And then underneath that, you'll find the rest of the epidermis, and then you will find the dermis underneath that. And here we got two scales on top of one another. This is the old skin that's being shed, and this is the new skin underneath. So reptiles continually grow throughout their life and shed their skin periodically as their body grows. This is a higher magnification of this slide over here, showing you old scale on top, new scale on the bottom. You can see here is the stratum corneum here, right? Here is the rest of the epidermis, and then the dermis underneath that. Here's the new scale underneath that old scale. Here's the epidermis that's associated with that, and the dermis that is alive still underneath. And these are all chromatophores or melanophores, all right? The melanocytes that produce the color that gives the snake's skin or the lizard skin its color. And this is the rest of the thicker dermis underneath. If you follow the scale out, it is going to fold underneath and form a hinge. That's what this picture is showing here is a hinge. Notice the hinge is highly folded. And basically what can happen is that hinge, the scale can spread out, particularly in snakes. When they swallow something that's much bigger than them, the scale can stretch. And that's what the hinges allow the scale to stretch out as they swallow things that are maybe twice the width of their body so that the skin doesn't tear. So that's what the hinges provide, the ability to stretch for snakes in particular when they're eating. Scales can also form ornamentation and be supported by bone or thicker keratinization, and they're called spikes or spines or crests in reptiles. And the stratum corneum is what's really waterproof and what's really hardened to form the scales in the reptilian skin. So as I mentioned, they shed their skin. So this is how shedding works. So again, these scales, for the most part, are part of the corneum, right? They're a corneal specialization. And so as the corneum forms into the scale, you'll have an old scale, and then underneath that a new scale will form. That is, some of the corneum on the deepest portion of the skin will thicken and form a new scale underneath the old. Where the new and old meet is called the stratum intermedium. And so when the old scale is been there for a while and the animal's grown a little bit and the new scale has formed underneath, the old scale will break off and shed away. The way we can separate these two is basically where the intermedium is, you'll get an invasion of white blood cells, particularly macrophages, and then fluid will accumulate. Basically lymph or interstitial fluid will accumulate in that area, and that fluid will separate the old skin from the new, and then you can shed it off. Some lizards and snakes shed their entire skin at once, or pieces of skin. The turtles can shed 1 or 2 or 3 scoots at a time on the outside. They're periodically shed as the animal grows. But that's kind of how ecdysis, the process of skin shedding, takes place in reptiles. Okay, bird skin. So bird skin is probably the one that's hardest to kind of visualize when you look at it under a histological section. You probably saw that if we looked at them in class. I think we did. All this feather is, and birds are covered in feathers, right? All the feather is a modified scale. It's a much more elaborate scale, if you will. And it's produced, again, by the epidermis. And I'll kind of go over how that happens in the next slide. But again, they have an epidermis and a dermis, and embedded in that epidermis and dermis are these incredible feathers. All right, so these are different pictures of feathers. Feathers start off really as a round, enclosed and sheath structure that's called a follicle, a bristle follicle, and inside the follicle is where the feather develops. When the feather is mature, it's called a pin feather, and it's held in the sheath really close to the surface of the skin. The sheath will break open and this feather emerges, if you will, and spreads out, and then will replace old feathers. The epidermis has similar layers in birds as it does in other tetrapods. It has a basale layer, a transitional layer, the middle layer or intermediate layer, and then the corneal layer. It's heavily keratinized because it's a reptile. Birds are reptiles, right? They do have some specialized glands in certain regions, like the uropygal gland, which is a gland that's associated with the tail and produces oils. And so you might watch a duck, for example, take its back legs and rub it against the tail and then rub its feet all over its body. And what it's doing is collecting oil from the uropygal gland and spreading it across its feathers to preen them. You'll see birds preen them. They also use their beaks to do this sometimes and spread oil across the feathers, which basically coats them, protects them, allows them to last longer, and also waterproofs them. So that's why preening is done and why grooming is done, to remove old feathers and to spread oil from the uropygal gland. Birds also have scales on their feet, some on the face, that are just reptilian epidermal scales. The dermis is made up of collagen, is very thick, has a large blood supply. Blood supply is needed to make those feathers. Also salt glands that are found near the eyes or within the nose or near the nose. Some of their ducts and some of the gland itself come from the epidermis. And then brooding patches are a specialized part of the skin that are invaded with blood vessels, and feathers are lost that send warm blood. They're found on the bellies of birds, on the ventral surface of the birds, and that's what they lay on or what they put the egg against in order to incubate the eggs or keep the eggs warm. There's a brooding patch on a robin. Here you can see it's a very swollen, enlarged area of blood vessels that's very warm, that incubates the eggs. This swelling of blood vessels and kind of thickening of the dermis is under hormonal control in the female bird. Okay, feathers. So feathers are really amazing. This is a cool picture of a young chick showing you the rows of feathers that develop. The feathers are highly organized in rows and how they develop along the body and wings. And that's why the feathers look so nice on the surface of a bird, because they come off of the skin in sections and rows of skin that are called pterali. The pterali basically organize how the feathers are going to come off of the skin. And you can see those really nicely on a newer chick that's eyes aren't open yet. All feathers, even though they vary greatly in what they look like as far as size, shape, height, and whatnot, are made the same way. They have a long shaft. The part of the shaft that's above the skin is called the rheios. The other part of the shaft is called the calamus, which is embedded in the skin. The fluffy part of the feather is called the vein, and the vein is made up of barbs that come off kind of at least a 45 to 90 degree angle, and they come off at regular intervals, and then they give rise to barbules that run parallel to the shaft, and these barbules have little hooklets on it that hook to each other to form a network that looks like this, right? And that's basically what feathers look like. So you got the shaft here, the rheios, and then these are the barbs coming off, right? And then the barbules go inside, and then there are little hooklets between those that link them together. And this gives the feathers its structure and its strength. This is a better picture of that if you want to see it. Feathers come in all kinds of shapes and sizes. So we got wing feathers, which are the hardest and produce lift. We got down feathers, which are more fluffy and help incubate and produce and hold in heat. You have tail feathers, which help turn the bird. You have contour feathers, which cover most of the body. And then you got semi-plume, bristles, and phyloplume feathers, which are involved in sexual selection and ornamentation, like what you see on a peacock, for example. There is even one bird that produces toxins or poisons that it covers its skin with, and those poison glands are epidermal derivatives. And it's called the hooded puidae. It's a really odd-looking bird. You can look it up if you want. Actually, it's a picture of one right here. Sorry. You can see it actually has warning colors here in orange. Looks like a little bit like an oriole. Okay, feathers. As I mentioned before, feathers develop like scales do. They start off as as a dermal papilla. It's a little swelling, if you will, that has blood vessels in it. And there'll be some follicular ridges that invaginate to kind of form a bump, if you will. And then this bump is going to be encapsulated and form a bunch of epidermal ridges within it. And these epidermal ridges then form into the barbs, barbules, and the shaft of the feather. And that whole thing is called a follicle because it's ensheathed in connective tissue. And it will, once it's developed, it's called a pin feather. And these pin feathers are really small, as you can see here. They're down here. As you can see, the feathers that opened up here. But on this chick here, you can see all these smaller feathers that look like they're compressed. They're still held in the sheath. Basically, what happens is the sheath breaks open, and then the feather spreads out, and will cover that part of the body just like the feathers above it have. So once the barbules and barbs and hooklets are all formed, and you're still in the sheath, it's called a pin feather. The sheath breaks open and releases the mature feather. That's kind of how it works. The feather, this production of the barbules and the keratinization and the mitotic ability of the dermis, of the dermal papilla to produce this, and to feed the mitotic ability of the epidermis to form all of these feather structures, is supplied by the dermal papilla, which the papilla has a really large capillary bed that's associated with it, and melanocytes that are associated with it to give color to the feather. The bump that forms here is called a primordium, and the primordium develops into the follicle, which is the feathers is sheathed in connective tissue in that follicle as it develops. Okay, so that's birds and their skin. Let's talk about mammals now. So mammals, the major derivative is hair, right? And we have a bunch of multicellular glands also that are associated with the skin. We have sweat glands in some mammals, and then we have wax glands are called cerumen glands. in some animals. We have cerumen glands. And then we have sebaceous glands, which produce oils. And then there are other specialized glands out there that you can find in certain mammals that are at least partly derived from the epidermis. So mammal skin has many more layers to it. It has not only the basale layer and the corneum layer, but now we have a spinosum, granulosum, and lucidum layer that's associated with the middle layers, or what we call transitional layers. The glands, we have sweat glands, sebaceous glands, scent glands, mammary glands. There are other specialized glands you can find in other mammals that are made by the epidermis. Hair is the major epidermal structure here that covers most mammalian bodies, right? The dermis is thick and it cements and holds these glands, blood vessels, and hairs. And all other structures like horns and antlers also can be at least partly cemented or held into place by the dermis. Glands are embedded in the dermis and produced by the epidermis. So they're epidermal derivatives, as are the hairs. Again, the cells of the epidermis are called keratinocytes, and the process of keratinization starts in the basale layer via mitosis, and as the cells move towards the surface, they are heavily keratinized or filled with keratin and other protein products that waterproof and harden the skin. Melanocytes, again, in most mammals are found in or around the basale layer and give color to the skin, and melanocytes are also concentrated in the dermal papilla, where the hair matrix is that makes up the hair follicle. We'll talk about hair structure here in a second. So here is a drawing of a hair and sagittal section, and here is a histological section. The hair are quite neat underneath a microscope. Basically, the part of the hair that protrudes past the skin is the cuticle, cortex, and medulla. So that's what you can pull on the outside of your skin is the cortex, cuticle, and medulla. The root sheets and the glass membrane are what embed or anchor the hair in place within the dermis. So this is you following the hair all the way down. Here is the dermal papilla, which is a concentration of cells that are undergoing mitosis. And then this is the blood vessels that supply the mitotic ability of the hair. And these cells are continually dividing and then organized and cemented into place to form the cuticle, cortex, and the medulla. Hairs grow. You cut them, they grow, right? You can see these layers a lot better in cross-section. So this is a really beautiful histological section of a hair in cross-section. So you cut the hair and then look down inside of it. And you can see here's the medulla in the middle, right? And then out on the outside of the medulla is going to be the cortex. And then on the outside of the cortex is the cuticle, which is a flat layer of cells. And that's what grows past the skin. The stuff that's embedded in the skin are these epidermal cells that kind of root the hair in place, and they're called the internal root sheath, the external root sheath, and then the glass membrane of that basale layer, or what is the basement membrane of the basale layer of those cells that are forming the root sheaths. Then outside of that is the dermis, the connective tissue that holds it into place. You have a muscle that runs next to it. That's called the erector pili muscle, which erects the hair, stands them up. And you have some multicellular glands that are associated with the hair. They're called sebaceous glands. Oops, that's the wrong way, isn't it? All right. So sebaceous glands, they dump into the hair follicle. So they're associated with the hair follicle. So up here is the epidermis, dermis. Here's the hair, right? This is the root sheath. Or the hair follicle, and then the hair extends, the medulla, the cortex, and the cuticle extends out past the skin. Sebaceous glands are found deep, and they dump into the hair follicle right here. And so sebaceous glands produce sebum, or what's called oil. The major hydrophobic molecule produced here, the major hydrocarbon here, fatty acid, is squalene. Right? So squalene is the major fatty acid that is produced that makes up oil. And oil glands, they waterproof the skin and lubricate the hairs, if you will, within the hair follicle. They can be quite abundant. They don't necessarily have to all be found with hair follicles, but most of them are associated with hair follicles. So they're much more abundant where their hair is. They're much less abundant where hair isn't. So palms, your palms don't get real oily, which is good, right? So that they're not slick. The heels of the feet do not get very oily because there's no hair there. So this is what sebaceous glands look like. They're lobular. And notice the cells that make up the glandular part of it are flocculent or bubbly. And they basically accumulate squalene. And they accumulate it at such a high rate that it eventually kills the cell. And the whole cell is released and breaks apart to form the thick oil. And this is called a holocrine gland, is the name given to it. Holocrine gland, because the whole cell is released. And so some of the cells continually divide by mitosis, differentiate, accumulate squalene, die, and break open in the lumen and release the oil and all of the cytoplasm and cellular contents into the hair follicle. Again, a nice picture here of a sebaceous gland, 2 major lobes. Here are all the cells. They divide from down here. They move up. They accumulate the white flocculent stuff, squalene, and then they break open and are dumped into the lumen of the hair. Pretty cool, right? All right. Okay, so let's look at some specializations here. So we talked about hair, how hair is formed, and all that hair is, as I mentioned, are keratinocytes that are kind of glued together and cemented in a certain way. They form the cuticle, the medulla, and the cortex. Color is added to the hair at the dermal papilla by melanocytes. But think about nails. Nails, claws, talons, hooves. So really big nail, actually. These, again, are just made of keratinocytes that are very, very keratinized and cemented really close together. And so the nails, you know, they break, they shed, they fall apart, they grow, right? Either cut them or bite them off. This is what the surface of a nail looks like, right here, or a hoof or a talon. These individual little flakes you're seeing, this is blown up under a scanning electron microscope, are keratinocytes cemented together. So there's a proliferating matrix, or a real high area of mitosis that gives rise to the underlying nail bed, and then all of these cells are basically flattened, heavily keratinized, and glued in place. And they can be very sharp, like the claws on a cat or whatnot, or they can be flat like ours, or they can be really big and thick, like the hooves on an ungulate, like a horse or a cow. So it's made up of very tightly packed, heavily cornified epidermal cells. So this is a cool picture. So this is a picture of a sagittal section through a rodent toe, end of a rodent toe. So here's the nail, right? The very flat nail, heavily keratinized. Here's the nail matrix, right, that is undergoing mitosis, and the nail plate, which cements the nail and grows the nail so that it continues to grow. You can see here's the epidermis coming over the top, forming a proximal fold. It's called the epinuchium. Here's hyponuchium. If you replace hypo with epi, the epinuchium, that's the cuticle on top here, the little half moon that you see on the nail. And then underneath is another cementing point here, which is called the hyponuchium. It's underneath the nail. Nuchium is nail. And then this is great. There's the nail bed and the nail. This is what's left over of the dermis. And this is actually bone right here. This is phalanx 1. This is a bone. And then this is the joint right here, awesome, of phalanx 2. Right? There are 2 to 3 bones in each of the fingers or toes. This is the foot pad. Right? Here's the foot pad here. Here's an eccrine gland that produces some oils and some other chemicals. And then you got a really thick heel here, which is thick epidermis. So that's a really nice image that shows you the nail matrix in particular, which is what gives rise to the nail. Some pictures, SEM pictures of the surface of a nail. Here's low power, here's high power. You can see the individual cells at the narrow edge that are making up the nail. They're just cornified keratinocytes. Again, this is a blow up of the nail plate, the nail bed, the epidermis underneath, and then the bone. Here's squamous epithelium, stratum corneum under the toe. So pretty cool. Nail plate, epidermis. Nice blood vessel right here. So again, these skin derivatives are just organizing the very heavily keratinized keratinocytes into these structures we call nails, hairs, horns, and whatnot. So there are tons of structures, right? Antlers and horns are another very good example where you combine bone with the skin. Antlers are temporary, horns are permanent structures that continue to grow throughout the life of the organism. So the horns on the rhinos, the horns here on the sheep. These are associated skins that you find in the bovidae groups, cattle, antelope, sheep, bison, wildebeest, rhinos. Antlers are shed, whereas the horns are retained year-round. Antlers grow from bone from the skull. Bone grows out and is supported. That growth is supported by this velvet, which is the thickened skin on top. The thickened skin on top has dermis that's highly vascularized with blood vessels, and that blood supplies the growing bone, which gives you the antler size and shape. And then towards the end of antler growth, getting close to the rutting season in the cervidae, the deer and the elk, the moose and caribou, this becomes very itchy, and they start rubbing it against things like fences and trees to get rid of the velvet. And then what's left over after you get all of the skin off of it is bone, right? The dead bone. And then these are shed after the rutting season. So pretty cool, right? Again, incredible structures that are put into place partly because the skin is highly mitotic and the growing bone underneath. We already talked about, for example, also turtle shells, right? But here's a really weird one. Baleen, or in the baleen whales, what's called whalebone, are actually not teeth. They are derivatives of the skin. So the largest of the whales are all planktonic eating whales. They eat what's in the plankton that's swimming around in the surface. Things like krill, shrimp, small fish are going to be filtered out of the water as these guys feed. They open their mouths, take in a big accumulation of water, and then they allow the water to run through the baleen, and they collect all of the krill, shrimp, crabs, whatever swimming around in the water that's small enough to fit through this curtain of baleen. All the baleen is heavily keratinized extensions of the roof of the mouth skin. And so they're formed into plates, baleen plates that have fringes at the end. You can see them in the whale. This is a cool picture here on the bottom of a skeleton of a baleen whale. This is the inside of the mouth. This is the upper jaw, the lower jaw. You can see the baleen growing down from the roof of the mouth. That forms this curtain that water would be moved through, and then you collect all of the krill on the inside of that and then swallow it. This is what a plate looks like. And basically what grows out here are the dermal papillae. So the papillae enlarge and become really long, and they form rows that form a plate. And basically the papillae, which is the dermis, the connective tissue of the dermis, and some blood vessels, is going to be capped with the keratin sheath of heavily keratinized keratinocyte cap, if you will, or sheath, that forms the bristle part, if you will, or the fringes, the frill, the featheliness, if you will, of the curtain that is very hard and collects food. And these wear down over time and have to be regrown. And so that's what baleen looks like. Pretty neat, right? Skin derivative. And then all kinds of thickenings of the skin, like rattles and snakes. The rattles of the snake is actually leftover rings of shed skin that they move around that make the rattle sound. Beaks are covered with a cornified skin, a heavily keratinized skin. Knee pads that you see in elbow pads that you see in camels. This is what they look like. Again, just to protect that joint, which they lay their body weight on. Apical pads, tori, which are thickenings of the skin, and friction pads that you see in primates and other animals are all basically epidermal thickenings and derivatives that kind of support and protect those areas. Calluses that you form on your skin are thickenings of the epidermis. All of those help protect your underlying structures or bone or skin in that areas, and those are often corneal thickenings that are happening there. Okay, I think that ends the skin. All right, so that's the lecture that we will use for this week. And I think I will start respiration next, or circulation, one or the other. So that will be the next lecture that goes up. This information will be on exam 3. I'm sorry, this information will be on exam 2, right? So we'll put this stuff on exam 2. So this is the last of the material for exam 2, and all of this has already been added to the review sheet for you. So have a great day.